Good day once again. So today we will be starting to discuss one of the biggest groups of microbes or bacteria. These are the Hemophilus and other fastidious gram-negative bacilli. So to start with, this group of microorganisms belong to the family Pasteurella. So this family Pasteurella includes Hemophilus and Pasteurella. So these are gram-negative coccobacilli. So you know for a fact when we pertain to coccobacilli, they are not perfectly rod or perfectly spherical organisms. Non-motile, they are aerobic. Some of them could be facultative anaerobic. They have the ability to convert nitrate to nitrite because of the presence of of course, you know this from the previous lesson, the nitrate reductase enzyme. They are oxidase positive and catalase positive. So this is the gram stain of the Hemophilus. Okay, two important species of Hemophilus. We have here the Hemophilus influenzae and para-influenzae. Okay, so hope you could see the morphology of the organism. Let me go back to the previous slide. Wait. Okay, so I hope you could see the morphology of the organisms. The organisms are actually coccobacilli. Okay, so do you know that these particular organisms are seen, okay, as commensals in the mucous membranes of the human respiratory tract. So some of the humans will not show, of course, some of us will not show the signs and symptoms because of the fact that our immune system are probably doing well. But once you become immunocompromised, so that's the time that these organisms could become opportunistic. It is also noteworthy to mention that these organisms could be very dangerous, particularly for neonates. Now, on the other hand, there is one interesting species here. This is the Haemophilus ducreyi. So this organism is the only species of Haemophilus that is sexually transmitted. So this one could cause the so-called STI or the sexually transmitted infection. So again, of course, they are pleomorphic. Let's review what we mean by pleomorphic. So the term pleomorphic refers to organisms that can assume a variety of shape. So not a perfect cocci, not a perfect bacilli. So that would make these organisms pleomorphic. Okay. So as you can see again, so here's an example of these coccobacilli. So some of them are coccobacilli, such as this one, and some of them are small rods, such as this. Okay, so the Haemophilus are obligate parasites of mucous membranes, or in previous text I've mentioned commensals. Okay, so 10 species are involved in human infections. So noteworthy to mention are Haemophilus influenzae, Hemolyticus, Egyptius, influenzae, and ducreyi. Now, you might be wondering, what do we mean by the term Haemophilus? Well, the literal translation of the Haemophilus means blood-loving. Okay, hemo refers to blood and philus refers to love of. Why? Because it's actually one of the few genera that would require two important factors, okay? They require X and B factors for growth. So, X factor, it's not the Cont'd X factor as you know it, okay? So, X factor is hemin or hematin. So, this is found in blood or in RBC. Okay? While on the other hand, V factor is a coenzyme known as the nicotinamide adenine dinucleotide or the NAD. So other organisms may require may supply V factor. So, such as S. aureus, one of the most commonly known organisms that would supply the V factor. Now, let me take note that not all species of Hemophilus would require both the X and V factor. So, for example, Hemophilus parainfluenzae would only require V factor. Okay? Because of the fact that they can produce X factor. Okay? Produces X factor, which would require the V factor. Okay. Forgive the tricycle for passing by. Okay, there you go. Now, Hemophilus parainfluenzae would only require V factor. Hemophilus influenzae would require both the X and V factor. Okay. So, Hemophilus, this is the example of Hemophilus, okay, as seen in 5% horse or rabbit blood agar. Okay, so Haemophilus hemolyticus species and sometimes Haemophilus ducreyi. Okay, so the problem with the blood agar plate is that it would only have the X factor. Okay, there's no V factor in the blood agar plate. Be it a horse or be it a rabbit blood agar plate, there's no V factor in there. Okay, so as what I've mentioned before, X factor is hemine. And the usual source of hemine is RBC or the blood. But since it doesn't require V factor, it may not support blood agar plate per se, may not totally support the growth of Haemophilus species that would require both the X and V factor. Okay, thus, it says here in the bullet, thus, some will not grow without the V factor added because the NADAs are present in blood agar plate, and this is the one that destroys the V factor. What is this NADAses? NADAses are enzymes that destroys the NAD or the nicotinamide adenine dinucleotide. Okay? Now, since blood agar plate would have an active enzyme, right? NADAs that would destroy the V factor. Therefore, blood agar plate would not be an excellent, okay? It will not be an excellent culture medium for the cultivation of Haemophilus species that would require both the X and V factor. Now, instead of using a blood agar plate, then it would be better if you will be using the chocolate agar. Why? If you recall, how would you prepare a chocolate agar? So remember, there's no Hershey's or Cadbury's in chocolate agar. Okay, that's a joke. Now, how would you prepare it? Diba you prepare it by heating the blood agar plate until such time that the blood, which is red in color, will turn brown, which appears like this one, a chocolate. So, since a chocolate agar plate is a heated blood, and heat destroys the enzymes, and in doing so, the heat destroys the NADAs. Therefore, chocolate agar would release the X and V factor and deactivates NADAs. Therefore, if you are using a chocolate agar plate, then it would be better in the sense that it would contain both the X and V factor. Okay, if you're only using blood agar plate, then blood agar plate would still have the active NADAs, an enzyme that degrades the NAD, which is the V factor. So, for you not to forget, okay, X factor and V factor. X factor is hemine. Okay? V factor is nicotinamide adenine dinucleotide. Okay. X factor is present only in blood agar plate and in chocolate agar plate. But V factor is present only in chocolate agar plate. It is not present in blood agar plate since the blood agar plate contain an enzyme that would destroy or degrade this NAD. Therefore, if the species of Hemophilus would require both the X and V factors, such as Hemophilus influenzae, using a blood agar plate would not be ideal. But if the species of Hemophilus would only require X factor, then you might as well use blood agar plate. Okay? So, however, in practice, chocolate agar plate, particularly those with trimethoprim antibiotics in order to serve as inhibitory agent, chocolate agar plate would be most ideal for Hemophilus species. Okay, I did mention a while ago that some organisms would require or would supply the V factor. Okay, remember that I did mention that a while ago. So, example of these organisms would include S. aureus, Staphylococcus aureus, Staphylococcus pneumoniae, Neisseria. Okay, so this species could cause hemolysis. Therefore, they could release the factors or naturally produce V factor. Okay, so why am I saying this? Because of this phenomenon would make it possible to have satellitism. Okay, satellitism or the so-called satellite phenomenon. Okay, so satellite phenomenon is the growth of fastidious organisms around other bacteria that release the necessary growth factors or break down the toxic products. Okay, or in other words, it is defined as the confluent growth of Hemophilus species, except for aphrophilus and ducreyi. Okay, in blood agar plate beside the colonies of S. aureus and S. pneumoniae or other organisms. So, just to make it clear, let's say, for example, this is your blood agar plate, and then you streak S. aureus. So, what will you have here? So, let's say, for example, now have the growth of Hemophilus colonies beside the colonies of S. aureus. What makes it satellite? It's because they're near from one another. The colonies of organisms are near from one another. So, what does it mean? Well, it means that since this is the blood agar plate, okay, supplies the X factor. The S aureus here, okay, the S aureus here would supply the V factor. Therefore, Hemophilus influenzae would be satisfied because here you have both the X and the V factors. Okay, did you get it? So this is what you call satellitism or the satellite phenomenon. Again, let me define this. It is the confluent growth of Hemophilus species beside the colonies of S. aureus or S. pneumoniae or Neisseria or any organisms that could provide the V factor. Common denominator of these organisms are capable of hemolysis that could release the V factor. Okay, so that's satellitism. Wait, I have a better picture. Okay, so again, this would be the blood agar plate. This would be the S. aureus. And the colonies that you could see here, the small pinpoint colonies, these would be the colonies of Hemophilus. Okay, so that's why it's called satellitism or the satellite phenomenon. Oh, here's another picture of that, a close-up view. So again, this would be the colonies of S. aureus, the bigger ones. Okay, and this would be the colonies of Hemophilus influenzae. Okay, do you know that 10% of normal flora of the upper respiratory tract, okay, is made up of Hemophilus species? About 2 to 6% in children from birth to childhood, with a higher percentage colonization in daycare center. So as they mature, they convert from non-encapsulated to from encapsulated strains to non-encapsulated strain. This is very important. You know why? Because the presence of capsule among the species, particularly of Haemophilus influenzae, okay, dictates the presence of virulence. You do not want to have your, let's say for example, you have a child in the future. You do not want him to have a colony or invasion of Haemophilus influenzae that are encapsulated. So let me say to you the fact that some strains of Haemophilus influenzae are encapsulated, and that spells virulence. Some strains of Haemophilus influenzae would be non-encapsulated. Okay, but you might be wondering, if the name of the species is influenzae, so it's like saying that these particular organisms could cause influenza. Now, okay, Haemophilus influenzae, okay, in contrary, will not cause influenza. Okay? So, historically speaking, in 1920s, during the Spanish flu, okay, the Spanish flu, the biggest flu of them all, H1N1 in 1920s. This particular flu is considered as the biggest because if we're going to sum up the total casualties of World War I and World War II, it wouldn't match with the casualties of H1N1. Okay, so H1N1 is caused by a virus. The name of the virus is orthomyxovirus. It is a type of RNA virus that has 8 segmented RNA genome, and because of that, this particular type of virus is capable of genetic reassortment, which means that it can combine, let's say, for example, bird flu, then swine flu. They would have genetic recombination, then the new strain would now be capable of infecting human. So you now have the H1N1. So there would be different combinations. Some would be, let's say for example, in 2005 in Vietnam, there was this H5N1. Now in 1920s, the biggest pandemic was H1N1. Now, as a result of being infected with viral infection, there's a chance that these people who got infected would have secondary bacterial infection. So, the secondary bacterial infection would most likely be caused by Hemophilus. Okay? But it is not really the Hemophilus that caused the H1N1. It's viral. Hemophilus is bacterial. So that's the reason why it was named as Haemophilus influenzae. So Haemophilus doesn't cause influenza. It just so happened that this type of bacteria is most commonly isolated from victims of influenza. Okay, because of secondary infections. I hope that's clear. Okay, so let's talk about the virulence factors. So, I told you that there are several virulence factors of Haemophilus influenzae. Okay? One of the most important virulence factor is the capsule. Take note that there are many serotypes of capsule, from capsule A until F. But the one that is most significant is the type B. Okay? The type B capsule. In fact, majority of the vaccine being developed against Haemophilus influenzae would act on type B Okay, capsule. So that's the reason why you might hear the term HIB. Okay, HIB refers to let's say for example, HIV vaccine. This refers to Hemophilus influenzae type B vaccine. Then of course, the IgA protease could cleave IgA in the mucous membrane. So that's the reason why majority of them are found in the mucous membrane. So other would include the lipopolysaccharide, though it is not well defined, and antibody to this protein is somewhat protective. The LPS would have the capability of paralyzing the cilia in the lungs. So that's the reason why there would be, of course, as you know, cilia propels mucus in the lungs, so you somehow can't clear the lungs if you paralyze the cilia. And then, of course, the adherence factors would include pili and other associated structures to it. Okay, so, but again, the most important virulence factor would be the capsule. So that's the reason why you do not want to have the encapsulated strain of Hemophilus influenzae. Okay, so I told you a while ago that capsule is the one that can prevent phagocytosis. And it's also anti-complementary, which means that it's against complement, and complement is actually needed in order to cause lysis of the bacteria. Primarily, it contains ribose, ribitol, and phosphate. So, as what I told you before, the HIV or the type B vaccine, capsule, is one of the most important since primarily cause disease in children. So, it is capable of causing bacteremia and it could spread to other tissues. So, that's the reason why HIV vaccine, as what I've explained to you, is useful in reducing incidence of the disease. So, there would also be non-typable strain, and that would also be possible if a certain strain of Hemophilus doesn't have a capsule. Though, the non-typable strain could invade the respiratory tract and any other surrounding tissues, which means that it could cause localized infections, such as pneumonia. Noteworthy to mention is this one, otitis media. This is the infection of the middle ear, and of course, sinusitis. Otitis media, most common etiologic agents of otitis media would include Hemophilus influenzae and Streptococcus pneumoniae. So, this is how the capsule would appear. Okay, so capsule surrounded the bacterial cell. 6 strains, but the type B is the one that could cause majority of the invasive disease. So I did mention already the composition, the PRP. Okay, let me go back. PRP, the ribitol phosphate. Okay, so PRP vaccine is also being given to reduce the incidence of meningitis and epiglottis. So, Hemophilus influenzae type B could cause meningitis, the infection of the meninges, epiglottitis, okay, septic arthritis, okay, cellulitis, and conjunctivitis. Okay, it can also cause the lower respiratory tract disease that may mimic influenza. Okay, so there's another one, another species of Hemophilus, Hemophilus ducreyi. This one is sexually transmitted. Okay, so as what I've told you before, the invasive type, okay, are the ones that could cause pediatric infection. So this would be the meningitis, cellulitis, epiglottitis. Take note of the spelling, kindly check this one. Then conjunctivitis. Those that are not encapsulated would cause mostly otitis media or the infection of middle ear. And for adult, particularly the capsulated strain, so this would result into pneumonia, bronchitis, sinusitis. So remember, as you grow older, the capsulated strain is converted to unencapsulated strain. So you do not want to possess, at your age right now, the capsulated strain of Hemophilus influenzae. So, meningitis is the invasion of the respiratory tract. Then what would happen is that the bacterium would allow the spread of the bacteria to the meninges and CSF. So mostly affected here are infants and children from 3 months to 6 years of age. Epiglottitis is, of course, the inflammation of the epiglottis. So there will be swelling and eventually obstruction of the respiratory passageway, which could affect children from 2 to 4 years of age. And if there is already a severe inflammation, then an emergency tracheostomy could be very helpful to relieve the condition of the patient. Bacterial tracheitis, well, this one is dangerous because it's life-threatening, okay? So this is the inflammation of the trachea, which is the windpipe, okay, responsible for normal breathing of children, or not only of children, of humans in general. But children are actually mostly affected with this one. Then cellulitis, the most common anatomic site for this would be the cheek. There would be swelling, pain, reddish blue color of the inflamed area. So children younger than 2 years of age could also be affected with this. So acute pharyngitis and pneumonia would also affect children about 1 year old children. Then otitis media, bronchitis, sinusitis. So just being mentioned again for you not to forget. Okay, so in summary, okay, study the table 18.1 of your book, 18-1 rather. This would be the different infections caused by Haemophilus influenzae as categorized into encapsulated and non-encapsulated strain. Okay, Haemophilus aegyptius, formerly known as Haemophilus aegypticus, but they actually changed the name to aegyptius. Okay, and Haemophilus influenzae, biogroup aegyptius. This one could cause conjunctivitis or the pink eye. So, you know, if you have sore eyes, there are two possible culprits. It could either be bacterial or viral. But in order to differentiate whether it's bacterial or viral, most of the time, if it's a bacterial conjunctivitis, okay, the type of conjunctivitis is purulent. Mostly, kumbaga sa Tagalog, mas mutain ka as compared to viral infection. Viral infection kasi ng sore eyes would most likely be reddish, inflamed, that's it. But it's not as purulent as compared to bacterial conjunctivitis. And it's not also true, for crying out loud, that if you've seen or if you look someone with sore eyes, you'll get sore eyes too. That's not true. That's the reason why wearing shades in order not to infect other is actually a misconception. Okay? Scratching your eyes. Okay? Using contaminated hand is the primary reason why you would have conjunctivitis. Therefore, if someone has a conjunctivitis, it's still okay to look at his eyes for as long as he won't touch anything, okay, for inanimated objects from that person. Let's say, for example, a contaminated handkerchief, or you don't borrow shades, or more so, contact lens from that infected person. Hey, why would you just borrow a contact lens anyway? So these are the different ways to prevent conjunctivitis. On the other hand, there is another biogroup, the Haemophilus influenzae, biogroup aegyptius. This, aside from conjunctivitis or pink eye, this one can cause the BPF, or the Brazilian purpuric fever, which mostly occurs in warm tropical climates. This one is kind of dangerous because of the fact that aside from the fact that you'll have recurrent conjunctivitis, you'll have high fever, vomiting, petechiae, purpura. This one is dangerous, septicemia, and then eventually your blood pressure goes down. You call that shock. It has a very high mortality rate, as high as 70%. Now, Hemophilus ducreyi here is an example of sexually transmitted infection. This is known as the chancroid or the soft chancre. Okay, so don't forget, soft chancre is caused by Hemophilus ducreyi. But then again, hard chancre is caused by syphilis, which is actually a sign of having syphilis, which is caused by another bacteria. This one is Treponema pallidum. Okay, so the incubation period of soft chancre or chancroid is 4 to 14 days. You'll have a painful lesion with an irregular edge. Okay, unlike Treponema pallidum, the lesion is not as painful according to the book, not according to the experience. Okay, so which areas could be affected? Of course, it could be genitalia or perianal areas if you have been involved with sodomy. So the genitalia could be at the penis or at the labia or the vagina. So there would be an enlarged and draining lymph nodes known as buboes. So Hemophilus ducreyi doesn't demonstrate the satellite phenomenon. Okay, so actually the organism appears like the school of fish. So this is how they describe it under the microscope. And here's an example of the chancroid. It's characterized by ulceration. Okay, by the ulceration in the genitalia with irregular edge. Okay, the Melkos test would be actually a diagnostic test. So mode of transmission is sexually transmitted. It's sexually transmitted. Direct contact with lesions would also be a possible route of transmission. School of fish, and this is how they describe it under the microscope. And the organism could be cultivated using chocolate agar plates supplemented with 1% isovitalex. Apparently, the school of fish here. Other species include the parainfluenzae, which could cause endocarditis, the Hemophilus aphrophilus, which could cause bite, wound infection, and endocarditis. Okay, so noteworthy to mention that Hemophilus parainfluenzae could cause the so-called hog influenza. Though it's a normal microbiota, it could still cause endocarditis, found in female genitourinary tract, and could be associated with vaginitis. So, specimen processing. Do you know that Hemophilus species would die rapidly? So, that's the reason why once you have the specimen, you have to plate it within 10 minutes so that you'd maximally recover whatever bacteria that are left in that particular sample. So, if you're collecting specimen for Hemophilus ducreyi, you have to clean first the specimen site with sterile saline or NSS and sterile gauze. So, you have to swab the base of the ulcer with cotton swab that has been moistened with sterile saline. Okay, then before you inoculate it in the plate. Okay, good luck to that. Okay, you have to touch the genitalia, of course, with your gloves. And then Hemophilus influenzae, you may use a chocolate agar plate, sometimes with bacitracin or sometimes with another antibiotics known as trimethoprim. So it helps inhibit other organisms, especially the respiratory flora. Okay, so what could be the possible specimen? It could be a nasopharyngeal swab, or it could be even CSF, particular if the clinicians are suspecting meningitis, or sputum for that matter. Okay, Haemophilus Egyptius, I did mention this a while ago, the 1% isovitalex that would be added to the chocolate agar plate. For ducreyi, the GC agar, okay, hemoglobin, FBS, vancomycin would be the composition. Now, very important to take note that they would require CO2, so candle jar would be ideal, and you might as well have the humidity chamber so that you would prevent drying of the plates or of the agar. Okay, so I think you did see it a while ago, the smear of Haemophilus influenzae showing cocobacilli. Okay, so besides chocolate agar plate, blood agar plate would also be ideal, but you have to remember this would only be ideal for organisms that do not require B factor, such as Hemophilus, aphrophilus. But if the organisms would require V factor, then do not use blood agar plate, use chocolate agar plate instead. McConkey agar, being a gram-negative organisms, Hemophilus could still grow there. So, the colonies are translucent, moist, smooth, with convex colonies. Mousy or bleach-like odor. But if you're talking about the encapsulated strains, because of the presence of PRP, the encapsulated strain colonies would appear to be more mucoid. Okay, so you know this already. Another picture of the Hemophilus influenzae. Okay, another colony and another colony of Hemophilus influenzae. Okay. So, in identifying Hemophilus influenzae, be sure that you take colonies from initial isolation. So, do not pick up the growth media. So, you place the colonies in nutrient broth and mix. Then, you plate inoculated nutrient broth on a nutrient agar plate or Mueller-Hinton agar plate. Okay? Then, you add X and V strips to the media. So, what is the purpose of this test? The purpose of adding X or V strips to the media is that you'll be able to know whether the colonies, or the bacteria would require X factor only, V factor only, or both. Okay? So, all of these plates should be incubated at 37 degrees with 5 to 10% CO2. So, probably in the CO2 packet or in the candle jar in 18 to 24 hours. Okay, so after 18 to 24 hours, you read the plates. Okay, so if there would be growth, okay, in both X and V factors, means that the organisms would require this hemine and NAD. Okay, if there would be growth on V factor only, but there's no growth on X factor, means that the organisms would require only the V factor. So as you can see, the colonies are only seen here in the V factor strip. Okay, now on the other hand, these organisms would only require the X factor. So organisms here do not grow. So, question, why is that in determining whether the organisms would require X or V factor, why is it that we're using either the MHA or MHA, not the blood agar plate? Answer, parang ako rin yung sumagot, diba? So, the answer is that if we're using blood agar plate, it will result into false positive. Why? Because the blood agar plate itself would already have the X factor. So, you won't even know whether the organisms would really require X factor or not. Okay, so it's more ideal to use a Mueller-Hinton agar plate. Now, if you don't have the X strips or the V strips, is there an alternative test? Yes. Okay, so for example, you have here the blood agar plate. Then you have here the chocolate agar plate. Okay. So, how would you know if the organisms would require the X and V factor? So, you have to answer this. I will not tell you the answer because I will just give it during the exams. How would you interpret if there is growth in both blood agar plate and chocolate agar plate? What do you think the organisms require? Blood agar X factor or V factor? What if there is a growth in blood agar plate but no growth in chocolate agar plate? And what if there is no growth in blood agar plate, but there is a growth in chocolate agar plate? So remember the principle, blood agar plate contains the X factor only. Okay? Chocolate agar plate would contain both the X factor and V factor. Now you have to interpret. Porphyrin test is another test. Okay? To determine if they don't require X factor. So remember that the porphyrin plus whether the porphyrin plus colonies of bacteria. Okay, so the principle is that the delta amino levulinic acid, or the ALA, gets converted to porphyrin or protoporphyrin. To know the result, you have to add a COVAX reagent. It's produced at the lower part of the tube. COVAX, or the Ehrlich reagent, which is made up of para-dimethylamino-benzaldehyde. And then, ideally, you should have a UV lamp, the Woods lamp, so that you'll be able to see a fluorescent reddish-orange under the ultraviolet light. Okay? So, if there is a fluorescent red-orange, such as this one, means that the organism here requires X factor. So this is a negative result, means that the organism here doesn't require the X factor. Okay, so you have to study table 18-2 in your book. So this would be the differential test for other species of Hemophilus and Aggregatibacter. Okay, so it will also tell you whether the organisms would require the X factor, the V factor only, the X, or both. Okay, so take note that this table is also very important because it will tell us which species of Hemophilus are hemolytic or not. So obviously, Hemophilus hemolyticus is hemolytic. Okay, so you have to study this table. So very important for identification. Okay. Another one would be the differential test for Hemophilus species. Now this time, Hemophilus influenzae are actually categorized into different biotype and the corresponding infections that they could most likely cause. So treatment, though we're not doctors, it's nice to know that there would be possible treatment. So cefotaxime, ceftriaxone would be the recommended treatment. Alternative treatment would include trimethoprim sulfamethoxazole, imipenem, ciprofloxacin, and chloramphenicol. Then for Hemophilus ducreyi infection, erythromycin could also be a very ideal treatment. Okay, so this group of organism is known as the HACEK. Okay, hindi ito yung hasik ng lagim, okay? HACEK refers to a collective group of organisms which include Hemophilus species, okay? Some would be, however, Hemophilus aphrophilus is now Aggregatibacter aphrophilus. A refers to Actinobacillus actinomycetem comitans. You have a tongue twister here. So now it is now known as the Aggregatibacter actinomycetem comitans. Then the Cardiobacterium hominis, and E represents Kingella corrodens, and K represents Kingella species. Okay. Generally speaking, we have actually discussed already the Hemophilus, but how about the other HACEK? Anyway, the organisms are gram-negative bacilli. They are capnophilic because they require CO2, and they could cause endocarditis. Now, the usual flora is the oral cavity. In fact, Kingella could cause periodontitis. Actinobacillus actinomycetem comitans could also cause juvenile periodontitis. Okay? So, thus, the pathogens are associated with human bite wounds causing septicemia and subacute endocarditis. How weird is that? Okay? So, it means that if you have HACEK in your oral cavity and you bit someone, okay, your bite could cause the death of that person, particularly if that person is immunocompromised, because the bacteria from your oral cavity would go to your circulatory system. You call that septicemia, and eventually the organism will go to your heart, okay, or to the heart of the person that you just bit. So it will result into subacute endocarditis. Okay, so the moral of the story: do not bite, particularly if the person that you're going to bite is immunocompromised. Okay, so tabulated herein would be the summary of key reactions and characteristics of HACEK and Capnocytophaga species. Okay, so let's discuss them one by one. So, Aggregatibacter aphrophilus, it means foam-loving. So, we have already described it as the organisms. Okay, so this is a picture of an Aggregatibacter aphrophilus isolated that is not X-factor dependent and is growing over the entire surface of a trypticus soy agar plate. So another example of Aggregatibacter that had grown in blood agar plate, and the general microscopic morphology of Aggregatibacter aphrophilus. Then Aggregatibacter actinomycetem comitans. Primarily, this is an animal pathogen, but could be isolated from the oral cavity of humans. Non-motile gram-negative coccobacilli, it would take at least 24 to 48 hours for them before they be able to see the distinctive star formation in the center of the colonies. Okay, I hope you could Google image search this very interesting distinctive star formation in the center of the colonies. Okay, so Aggregatibacter actinomycetem comitans could ferment carbohydrates in the presence of serum, though lactose or sucrose, okay, are not fermented. So they are catalase positive, oxidase variable, but interestingly, even if they are gram-negative, they couldn't grow in McConkey agar. So they are also urease, indole, esculin, and citrate negative. So here's the photograph of Aggregatibacter actinomycetem comitans, formerly known as Actinobacillus actinomycetem comitans. Okay, so here's the blood agar plate. This one is the star-shaped centers of the colonies that are not usually evident after 48 hours of incubation and are best observed using 100 times magnification or stereo microscope, which means, guys, you cannot see the star shape here because this photograph is not under the stereo microscope. But if you will be placing this blood agar plate under the stereo microscope, then you'll be able to see the star-shaped colonies. Then the Cardiobacterium hominis is another pleomorphic non-motile gram-negative bacillus. Sometimes appears false positive on gram stain, okay, but they are really indeed gram-negative. So they have this capability of showing rosette morphology, and this Cardiobacterium hominis, okay, is a normal microbiota of nose, mouth, and throat. So, Cardiobacterium, perhaps the name Cardiobacterium refers to the fact that this organism could cause endocarditis, which of course involves the heart valve and is resistant to many antibiotics. So, as a result, it's very expensive. The valve must be replaced. So, the organisms could grow in blood agar plate, chocolate agar plate, but then again, not in McConkey agar. So, pitting may be produced on agar plates. Okay, so this is the 48-hour growth of Cardiobacterium hominis. So, here's the gram stain of Cardiobacterium hominis. So, what could you say? You have here the typical rosette formation. Okay. And then, Kingella corrodens. Normal flora of oral and bowel cavities. So, this is the fight and bite wounds. Imagine, no? If you're fighting with someone, then try to bite them. You watch out, I have Kingella corrodens. Okay. So, what will happen? Infections. Okay. Could result into infections. But not necessarily the bite, but generally speaking, Kingella could cause meningitis, pneumonia, osteomyelitis, arthritis, post-operative tissue infections, usually from bacteremia associated with wounds, particularly if you are not taking care of wounds or if a person is diabetic. Okay? Could also cause cellulitis, inoculation with needles lick clean, the so-called needles lick clean. So, Kingella should grow in environment that is rich in carbon dioxide. So, this one requires hemine or the X factor. Okay? And it is a gram-negative Coccobacilli. And once you are culturing it, you'll be able to smell the bleach-like odor with yellow colonies pitting the agar. So, that's the reason why it's called corrodens, because of the pitting. So, biochemical tests would also be helpful in identification of Kingella. It is non-motile, non-fermenting, oxidase positive, catalase negative, positive in lysine decarboxylase and in ornithine decarboxylase, but negative in arginine decarboxylation. They just summarize it here. So, here's another pitting in the colonies of Kingella. Okay. And the gram stain morphology of Kingella. They are much bigger than Hemophilus. And then, of course, we have here the Kingella. This is a Coccobacilli in short rods and in pairs or chains. They are non-motiles but may twitch. So, this organism is oxidase positive, catalase negative, and they are sugar fermenters. So, Kingella species include the dentrificans, Kingella dentrificans, which could grow on tire martin medium that resemble Neisseria gonorrhoeae, except that here, Kingella species, the colonies do not pit. So, there are gram-negative rods that have square ends. So, biochemical tests include a positive fermentation in glucose and positive reaction in nitrate reduction, but negative in urease, indole, esculin, gelatin, and citrate. So, Kingella kingae is isolated in children, particularly those who are below 5 years. So, they cannot ferment sucrose, but could ferment glucose and maltose. So, here is an example of Kingella with square ends. I hope you could see here the species of Kingella with square ends. Okay, Capnocytophaga. Previously called the DF1, DF2, which refers that they are dysgonic fermenters, which means they cannot ferment, okay, as much as compared to other bacteria. That's why the term dysgonic. So, gram-negative bacilli often fusiform. So, that's the reason why they may resemble the Fusobacterium. So, they are anaerobic, but however, they are also capnophilic since they require carbon dioxide for growth. So, the organisms could cause septicemia, okay, more common in cases of septicemia rather than endocarditis, but they could still cause endocarditis for that matter. So, here would be the growth of Capnocytophaga on chocolate agar plate. So, remember, take note that, for example, here's the colonies, but the organism could spread further. Okay? So, notice the spreading away from the center of colonies Okay, so you might compare this one with Kingella. But the pitting of Kingella is actually straight, more straight as compared to Capnocytophaga. Okay, so here's another gram stain photograph of Capnocytophaga. And this is an advanced periodontitis, which is different from dental caries because in periodontitis, even the surrounding gums, okay, aside from the teeth, the gums, and the alveolar, the bone deeper here, okay, are infected. So, Capnocytophaga. So, hindi lang ito yung pwede mag-cause ng periodontitis. Kingella could cause. Actinobacillus actinomycetem comitans could also cause periodontitis, which is much severe than dental caries. Okay, so Pasteurella C would be another organisms. Under Pasteurella C, we have the Pasteurella multocida, the most commonly encountered species, since these organisms are part of upper respiratory tract flora of dogs and cats. Pasteurella species will grow on blood agar plate or chocolate agar, but not on McConkey agar. They are small, gram-negative bacilli that are oxidase positive, and contrary to most other gram-negative bacilli, they are sensitive to penicillin. Now, this one is unique because most of gram-negative bacilli would be resistant to penicillin. So these organisms are not particularly virulent and cannot establish infections unless exposure to animals allows entry, mostly by means of animal scratch or animal bite. So there are about 5 serogroups of Pasteurella multocida. A, B, C, D, E, F. I think that's 6. Anyway, gram-negative pleomorphic coccobacilli with bipolar staining. So again, the term pleomorphic means that the organisms could assume a variety of shape. So oval shape, some are short rods, some would have a longer filament. So the organisms could grow on blood agar plate and chocolate agar. So they are non-hemolytic mucoid colonies with a narrow green or brown halo formation. So most infections are localized to the point of entry, particularly where animal bites had taken or where you have been bitten by an animal. So you are more predisposed to have infection if you are immunocompromised. Okay, so the joints, the respiratory tract, the central nervous system could be affected. Pasteurella multocida could colonize the upper respiratory tract and the GI tract of mammals and birds. So exposure from dog and cat bites or scratches, okay, would be the most important predisposing factor to have this kind of infection. So the biochemical tests, they are non-motile, catalase positive, oxidase positive, but could weakly ferment glucose because of the weak acid formation. Okay, so summarize herein in table 18-5 would be the differential characteristics of Pasteurella species associated with human infections. So you may read it in your book. And this would be the colonies of McConkey, or rather of Pasteurella multocida, as shown in blood agar plate, McConkey agar, and in chocolate agar plate. So take note that Pasteurella is positive, okay, in blood agar plate and chocolate agar plate, but the growth is negative in McConkey. Okay, so that would end the screencast. We will just continue I will just have another file for the continuation of Hemophilus or for other members of fastidious organisms. Okay, ciao.